Hello, welcome back to AE course. In previous session, we talked about passive transport as well as simple diffusion, osmosis, and facilitated diffusion. Now, in this lecture, we will focus on the active transport. We will talk about primary active transport, secondary active transport, as well as vesicular active transport. So, first of all, let's see what is the meaning of active transport. I talked and I mentioned in the previous session that we have two types of transport, either passive or active. This active transport, it means transport of the particles against their concentration gradient from the region that has low concentration of those particles to the region that has higher concentration of that particle. For example, imagine you are in the, again, like the other examples, imagine you are in the metro and the train it's full of the people and you want to get into that train. You spend a lot of energy to get in that train. It's exactly like that. For example, so you have to spend energy and go from the lower concentration to the high concentration. In other words, you go uphill. It is the uphill movement. And in this type of transport, you also need a carrier protein. Carrier protein help and facilitate the movement of these particles through the membrane. And these carrier proteins, they are highly specific. They can only bind to those ligands that are related to. For example, as you see here, there is sodium and potassium pump and they will only bind to sodium ion and potassium ion and transport these type of ions, not any others. So, with this introduction about active transport, let's see what is primary active transport and then secondary as well as vesicular transport. So, what is primary active transport? Primary active transport, as you see, it is active. So again, it needs energy. First of all, you must know that it needs energy. And primary active transport is that type of active transport that during which carrier protein will use ATP directly to transport particles against their concentration gradient. So they will degrade ATP into the ADP, and that inorganic phosphates will bind to this carrier protein and change the shape of the protein and facilitate the uh, transport of the particle through the cell membrane. So the main thing about primary active transport, now we know that directly use ATP and it's again against their concentration. So directly use ATP and transport those particles against their concentration gradient. As you see here, it is a sodium potassium pump. It will transport three sodium out and transport two potassium inside of the membrane into the intercellular fluid. And there are some examples for primary active transport. It's good to mention like sodium, potassium pump, calcium pump, and proton pump or hydrogen ion pump. In the primary active transport, these carrier protein, they also act as an ATP degrading enzyme. As I mentioned, they will change ATP into the ATP. So they are ATP degrading enzyme. And in this type of transport, you see that two types of particles transport through the membrane. So we call it co-transport. It's co-transport because two types of particles transport through the membrane. And why it's antiport? It's antiport because these particles they transport in the opposite direction. One of them, which is for example potassium ion, move inside, and the sodium ion move outside of the cell. So their direction is opposite. That was all about primary active transport. Let's see what is secondary active transport. Secondary active transport, it's also active again, so it needs ATP and also transport particle against their concentration gradients. And so when I say against, it means from low concentration to high concentration gradients of that particles. But there is a main difference. There is a main difference between the secondary active transport and primary active transport, and that difference is these carrier protein that transport particle against its concentration gradient will not directly use ATP. It will use the energy of the concentration of the another particles. For example, in this diagram, you can see here in this picture, in this diagram, this protein, use the energy that's stored in concentration gradient of the sodium and then uh, transport glucose to the fluid, intercellular fluid. So what will happen in the secondary active transport? In secondary active transport, as you see here, there is a protein here. It will produce concentration gradients and also use ATP. So ATP used with this protein, and it will make concentration gradient. And the other protein, which is here, you can see, which is here, and this protein, as the sodium ion pass through this protein, it will release energy, and this protein use that energy to transport glucose, for example, to the inside of the cell. So another time, let me talk about it another time. So one protein in the other side will use ATP, and another protein, another carrier protein, use the energy that is stored in particle concentration gradient and transport another particle. So in secondary active transport, it will always transport two particles. One of them will transport down its concentration. It means from high concentration to the low concentration. As you see here, sodium transport down its concentration gradient, and glucose or the other particle always transport against its concentration gradients. And this secondary active transport, there are mainly two examples for them. It's sodium ion glucose symport transport of them, and amino acid sodium ion antiport system. What is the meaning of symport? Antiport, I said, what is antiport? Let's see what is symport. Symport is another type of co-transport. It means two types of particle transport with the carrier protein. But in this transport, these two particles, they transport in the same direction. In the same direction. That's all. Let's see what is vesicular transport. The other type of transport which is also use energy, it's vesicular transport. Vesicular transport, vesicular transport can be either intracellular or intracellular, which means inside of the cells, those transport of the vesicles inside of the cell within the organelles, and intercellular, it means between the cells. When it's inter, it means between the cells. When it's intra, it means inside of the cells. In the intracellular, I don't want to talk about it very much, but in the intercellular, for example, for transport of those large protein or those polysaccharide inside of the cell, they use vesicles, and these will be the intracellular vesicular transport of the cell. But intercellular, between the cells and the extracellular fluid, it can be either exocytosis or endocytosis. So intercellular vesicular transport, it can be either exocytosis or endocytosis. What will happen in the exocytosis? In the exocytosis, as you see here, those protein or those particles that are produced by the cells, for example, like insulin, which produced by those ribosomes which are located on the rough endoplasmic reticulum, those bonded ribosome. And then these protein they packed in the vesicles, and then these vesicles, which are come from Golgi apertures, they fuse with the plasma membrane, plasma membrane, and diffuse those particles out of the cell. That is exocytosis, for example, for secretion of insulin in the, for example, insulin in the pancreas. They use this type of transport. And let's see endocytosis. Endocytosis is also another type of intercellular vesicular transport. In the endocytosis, those large particles, such as, again, it can be protein, fibers, those dead cells, or anything that will come into the cells and then ingested with these vesicles. As lysosome can bind and secrete its enzymes to these vesicles and digest those particles which are inside of the vesicle. What will happen in the endocytosis? In the endocytosis, those particles will reach to the plasma membrane, and then a small part of the plasma membrane enclose those particles. You see here, it will step by step in this direction, and it will taken up those particles into the cells, and after that these particles ingested with the cells. They can be fibers, again, anything. uh, White blood cells like macrophages, neutrophil, they can have endocytosis, and endocytosis can be two types. As you see here, they can be two types. Based on the size of the vesicle, there are two types. There are either pinocytosis or phagocytosis. Pinocytosis, when the size of the vesicle is small, we call it pinocytosis. It's also called cellular drinking because those fluids and solutes ingested in these with this type of endocytosis. And phagocytosis, it's also called cellular eating. It consists of those large vesicles, and it will ingest those large particles such as fibers or invaders, pathogen, and these type of particles. And that's all about the movement through the cell membrane. And I hope in the next episode, we will talk about cell junction between the cells.